It's Wednesday, so which means we have our last book talk of the calendar year, right? We're up to part 4 of the Hot Zone, and it's also game day, which means during this period, after you do the Bellringer, after you do that puzzle, and after you do the book talk, I'm gonna sit down and start playing some of your games, both today and tomorrow, right? So I'll play most of the games on whatever day is your lab period, today or tomorrow. And not only that, we're getting down to the nitty gritty when it comes to the countdown. We're up to number 2 on the countdown. That's right, number 2, dose, number 2, which is... All right. Hey now. Mr. Kovacs class. It's Mr. Kovacs class. You might learn on time. It's Mr. Kovacs class.
It's interesting too.
It's Mr. Kovacs class.
And he's super cool.
It's Mr. Kovacs class. That's right. Can't live without him. The cell membrane. Cell membrane is number 2 on the countdown. Love the cell membrane, right? It's hydrophobic. Right, it keeps the inside in, the outside out. We're for cell membranes, we'd just be puddles on the floor. And cell membranes are the key component, probably the most important part of a cell that there is. So let's look at cell membranes again. That's number 2 on our list. Remember our friendly old fluid mosaic model of the cell membrane, right? You remember that? Fluid mosaic model, right? Fluid, because it's constantly in motion. You see these phospholipids with their hydrophobic heads and their hydrophilic tails. They line up in a bilayer, right? That's fluid. And then they have all kinds of like proteins and things and cholesterol and other things floating around in them. So if you look at it, it's a mosaic. So we call it the fluid mosaic model of the cell membrane. And then we talk to cell membrane controls what goes in and out of the cell, right? Like sometimes cells do what's called endocytosis, where they just engulf particles by wrapping their cell membrane around the particles, right? Endocytosis. Sometimes they do exocytosis, like when they make things and need to get rid of them, they wrap them in a membrane and send them to the, right? This only works because the membrane is fluid. And we learned that there's 3 basic ways that the particles can move through this membrane. Since this membrane can control what goes in and out, we call it selectively permeable or semi-permeable. And if you remember the 3 ways, 2 of which do not require energy at all. The 3rd one requires energy. First one is just simple diffusion, right? That's when particles move from areas of high concentration down a concentration gradient until you have equal concentration on both sides of the cell membrane. Simple diffusion doesn't take any energy whatsoever. Diffusion. Facilitated diffusion is when the same thing happens, except for like small, like large things or things that are polar, like water molecules can't get through the membrane, so they have to travel through a channel. That's facilitated diffusion. They travel through a cell membrane protein, either channel or carrier protein, to move from areas of high concentration to areas of low concentration, right? Water does this famously. Water moves through aquaporons, which allow it to move from areas of high concentration to areas of low concentration through diffusion. And of course, we learned the diffusion of water. It's got a special name. What's that name again? That's right, it's called osmosis. Right, and then what's the 3rd type? The 3rd type is when you use energy. That's called active transport. Use energy to push things across the concentration gradient from areas of low concentration to areas of high concentration. You can push them against the grain. That requires energy. And the energy that they use, of course, is our old friend from the mitochondria, ATP. Right, so cell membrane we learned, right? We've got simple diffusion, things moving through the membrane from high concentration to low concentration. We have facilitated diffusion, things moving from low concentration to high concentration, but through a channel. Then we have active transport, right, which is where you go against the concentration gradient, but you need to use energy to do that. Right, the cell membrane. Of course, we did that lab with cell membranes, right? If you remember the lab where we took a cup, making a mess over here, but you made a fake cell, right? And you filled it with starch and glucose solution. You stuck it in some water, you put iodine in the water, and the iodine moved by diffusion into the cell and turned it that blue-black color, right? And then you tested the water on the outside of the cell, and you found that sugar, indeed glucose, moved out of the cell because it tested positive with Benedict's test. That's always on the Regents, questions about that diffusion lab. All right, so now that you're done with today's Bellringer, you're gonna scoot along and do the Ed puzzle about cell membrane, and then you're gonna do the book talk about part 4, and then you're gonna get together and I'm gonna start playing your games. Yeah, today's the big day, today and tomorrow. All right, let's do it.